Children, welcome to biology class. Today, again, we'll continue chapter Biotechnology, Principles and Processes. So, today we'll learn about the tools of recombinant DNA technology. So, the tools which are required for recombinant DNA technology, they are the enzymes, cloning vector, foreign DNA, and competent host. So, let's come to enzymes. So, enzymes which are needed for recombinant DNA technology are of three types: lysing enzymes, cleaving enzymes, and synthesizing enzymes. So, lysing enzymes are used to open the cells or to dissolve the cell wall of plant cells. Usually, lysosomes are used to dissolve bacterial cell wall. Cleaving enzymes are used to cleave a DNA molecule into segments or to open a circular plasmid DNA. Now these enzymes, they are called nucleases and they are of three types: exonucleases, endonucleases, and restriction endonucleases. So exonucleases, they remove the nucleotides from the 5 or 3 end of DNA molecule. Endonucleases, they cleave the DNA duplex at any point within but not at the end, and they cleave only one DNA strand. Restriction endonucleases are the nucleases that cleave both the polynucleotide strands of DNA at a specific sequence of nitrogenous base. Now these specific sequences, they are called recognition sequences, recognition sites, restriction sites, or target sites. The recognition sites, they have palindromic sequences of nitrogenous bases. There are specific restriction endonucleases for specific palindromics. For example, restriction endonuclease 1 recognizes palindromic sequence GAATTC in both strands of DNA in 5 to 3 direction. So here children see the exonuclease, endonuclease, and restriction endonuclease. So these are the two strands of DNA. So exonuclease, it can cleave the strand at the end of 5 or 3 strand. Then endonuclease, it cleave the DNA on molecule of only one strand in between. And restriction endonuclease, it can remove the polynucleotide strand of DNA of nitrogenous bases at specific points, which has palindromic sequence. Now restriction endonucleases, they are basically produced by many bacteria to destroy the bacteriophage DNA that happens to enter the bacterial cell. Now these enzymes, they protect bacteria against phage infection. They are called restriction enzymes because they restrict the multiplication of phages and cut DNA strand only at restriction sites. So here it is a bacterial cell. This is a bacterial cell, and this is a bacteria DNA, which is as a host. Now this is a phage, and this restriction enzyme, it cleaves the incoming phage DNA at recognition sites into fragments. Now functioning of restriction endonuclease. Now each restriction endonuclease, it recognizes a specific palindromic base sequence in foreign DNA and cuts sugar phosphate backbone of both the strands of double helix at specific points in the palindromic site. Now it exposes the single-stranded free ends from each fragment of DNA duplex, and these overhanging single-stranded free ends of DNA, they are called sticky ends. Now DNA molecules, they cut by the same restriction endonuclease which have the same sticky ends. Because this restriction endonuclease, it acts as a molecular scissor, and they have the same sticky ends, and they have similar complementary ends of DNA fragment from the same or some different source they can join. So here the formation of recombinant DNA by the joining of sticky ends of two different DNA molecules which are caused by the same restriction endonucleases or EcoRI. So here this is the vector DNA and foreign DNA. So here the restriction enzymes, it cuts the particular site and they both have the sticky ends. Then these ends, DNA fragments, they join at the sticky ends and forming the recombinant DNA. Now synthesizing enzymes, they help in synthesis of DNA, and they are the DNA polymerases, reverse transcriptases, ligases, and alkaline phosphatases. DNA polymerases enzymes help in synthesis of complementary DNA strands on DNA template. Reverse transcriptases help in synthesis of complementary DNA strand on RNA template. And ligases, they join or seal the gaps between the DNA fragments. And alkaline phosphatases removes phosphate group from 5' end of polynucleotide strand. Next tool is cloning vehicles or vectors. We'll learn in our next class. Till then, thank you and have a nice day.